डियर स्टूडेंट्स, जैसा कि मैंने अपने पहले लेक्चर में बताया था कि हम KBT 503 एकेटिव सिलेबस के हिसाब से अपना पूरा सिलेबस कंप्लीट करेंगे। इन कंटिन्यूएशन ऑफ दैट, टुडे आई एम गोइंग टू स्टार्ट विद द फर्स्ट यूनिट, दैट इज बायोलॉजिकल डेटाबेस. So before starting about the biological databases, we must understand what is bioinformatics. So as you can see here, I have described the bioinformatics with the different colors. So each color has a one meaning. So यहाँ पे आप देख सकते हैं कि we have different branches which are included in the bioinformatics. The very first one is a biology. Second one is information, means it is information technology, IT, mathematics, statistics, and computer science. So combination of all these fields becomes a bioinformatics. तो हमें इसकी क्या क्या ज़रूरत पड़ती है, जैसे कि वी हैव अ सीक्वेंस, जैसा कि हम डेटाबेसेस में डिस्कशन करेंगे. सपोज वी हैव अ DNA सीक्वेंस. So what you can infer from this DNA sequence, what are the informations you can collect from the sequence? For that you required various tools and various programmings, okay? And then you will make some assumptions. So hypothesis, you required some statistical data, some calculations you required mathematics. IT deals with the infrastructure, like we required computer, okay, and the technology what they are using, and the computer science means with the programming. So all the fields combine together and make bioinformatics. So अगर हम इसको definition में define करना चाहें, so we can define bioinformatics as study of the techniques and methods of molecular biology. That is very important. By the means of computational methods defined as bioinformatics. So हम वो सारी molecular biology techniques जो हम अपनी लैब में परफॉर्म करते हैं, अगर उन्हीं टेक्निक्स को हम कंप्यूटर की मदद से सॉल्व करें with the help of computer, so that field is known as a bioinformatics. Let's first talk about the databases. What is databases? Data refers to the piece of useful information. That is very important. We can have lots of information, but the useful information is referred to the data, while basis is the place where we can store the important information. So combination of that, the storage of the useful information. So we can say storage of useful information, that is database. So a place for the storage of useful information in a defined manner. Defined manner means we must store data in a proper manner by which we can retrieve the data when required. If we'll just throw the data into the databases and we will not have any format, so it is very difficult to get the data back when we require. Okay, so storage of useful information in a defined manner could be referred as database. If the data that database contain biological information, it is known as a biological database. So biological database we can define as a biological data are intricate, extensive, riddled with exceptions, and unfinished. What is the meaning of unfinished? Suppose you have sequenced a complete DNA sequence. So it is around the size of is 3 into 10 to the power 9 base pairs, approximately, if it is a human DNA. So in that case, what the information you will get it, because as all we know that the complete DNA do not code for the protein, so that is around only 2 to 3%. So from this 2 to 3%, how you will identify from this to this 3 into 10 to the power 9 base pair, it is 9 base pair, how you will get the 2.3% that is coding. So that's why it is unfinished. So the data which is useful for our purpose, we have to find out the data. Okay, so that's why it is unfinished. So we have to make the sense from this whole data. Okay, so to provide clear findings, a number of databases have been constructed and analyzed. A biological database is a collection of biological data organized in a way that is computer readable, quick to search and retrieve, and simple to use. A good database has to have the current data. So if suppose it is 2023 and a database have the 2000 data, then it means that database is not updating themselves. So we do not look into that data. We require the updated data, okay, for any kind of research. What is the importance of biological database? So biological database may be used to obtain a variety of data including biological sequences, structure binding sites, metabolic connections, molecular actions, functional correlations, protein families, motifs and homologs. आपकी इसी यूनिट में ये जो कुछ टॉपिक्स टर्म्स हैं इसमें we will discuss when we will reach there. Okay. A biological database is a primary function to store and manage biological data and information in formats that computer can understand. So as I do, very much focusing on formats. So there is a one next topic is molecular file formats. So I will see that I will discuss each file which is of your interest. We will discuss about it when we will reach there. Now these are the different databases. I have prepared a chart over here. So here I have divided a database into the three different categories: primary databases, secondary databases, and composite databases. We'll see one by one. Okay. So primary database are nothing but they are the only sequence database. Sequence means suppose you have a sequence like A, T, C, G, A, G, A, C. So this is sequence. There is nothing else. But from this sequence itself, we can get lots of information. I'll show you on just when we'll go one by one how we can infer the information from this sequence itself in a from particular database. Okay, so we will see. So primary database only contains the sequence. Either it is a DNA sequence, it is RNA sequence, it is protein sequence, or whatever it is. It just contain about the sequence, nothing else. So primary means it's a basic databases which contains the sequence. So there are two different types of sequence database, that is nucleotide and the protein database. So nucleotide database contain the main databases for nucleotides are the GenBank, EMBL and DDBJ. Before I come here, I just discuss a little about the history of the databases. What is the requirement of the database? Why we need databases? Okay, so in the early uh, 80s, when Margaret Dayhoff, Margaret Dayhoff, she is known as a father of bioinformatics. You can also say mother, but she is father of bioinformatics. When she started working on the proteins and she started getting the sequence, and she faced lots of problem like how we can store it, this sequence which I have received or which I have identified. Then she got an idea to develop a database, and she made the very first database, and that is a protein database, that is PIR, which is mentioned here. It is a protein information resource. Protein information resource. So this was the first database which is made by Margaret Ahab. Okay. Okay, again come back to the nucleotide. So there is a three databases, that is GenBank, EMBL and DDBJ. GenBank is a part of NCBI and it is defined as a gene bank. Gene bank. EMBL stand for European Molecular Biology Laboratory. DDBJ stand for DNA Data Bank of Japan. Okay, so it is from name itself, it is very much clear. It is maintained by EMBL, EBI, and it is maintained by DDBJ, like DNA Data Bank of Japan. It is from a Japan data, it is European data, and it is US based, USA US data. Okay, so these database contains a different sequences when they were just started. In NCBI is developed in 1988, and approximately the established time is almost same for all the database. Okay, so what was the problem is like this database only contains the information about the USA data. So suppose if we want to get the data from Japan, then I cannot get it from here. We have to go to the DDBJ database. So then they made a collaboration, like we must share our data from one particular source. We can get all kind of data. So they made a one collaboration, and that is INSDC. INSDC is DC is stand for international. It is very much important for 2 marks question. International nucleotide sequence database collaboration. Okay, so this is very much important about it. Okay, so what actually INSDC does is, at every evening they all three sit together and they share the data, and all the data is the part of INSDC. So any data you can access from DDBJ, you can access the data which is present in Gen from which is present in GenBank. You can access that data which is in EMBL. So everything you can do it from INSDC. Okay. So in databases, there is nothing to learn from your side. You have to understand the basic concepts of database, what that particular data does, that database does. Okay. So for that, I have given the link for each and every database. Okay. You can go to that particular link and you can identify. I'll show you just now. Okay, just finish this part. So suppose if it is a GenBank database, I'll just click over here and I have opened the GenBank, so you can see here what is GenBank. GenBank is a NIH genetic sequence database, an annotated collection of, what is annotated means identified, collection of all publicly available DNA sequences. GenBank is a part of International Nucleotide Sequence Database collaboration, as I told you just now, which comprises the DNA Data Bank of Japan, European Nucleotide Archive ENA, and GenBank at NCBI. These three organizations exchange data on a daily basis. So, a GenBank release occurs every 2 months and is available from the FTP site. So, what is FTP site? It's a flat file format site. Okay, how the data looks like. Okay, and so this is data uses, data processing, status and release, confidentiality, privacy, everything you can find it over here. Okay, so like that only it is EMBL. Let's see about the EMBL. The European Nucleotide Archive. Okay, European Nucleotide Archive provides a comprehensive record of the world's nucleotide sequence information. But when it was established, it was just a European nucleotide sequences, covering the raw sequence data, sequence assembly information, and functional notation. Okay, so this is the basic information available here. If you go to the about section, you can get the more information about it. Okay, for your writing part of purpose. Okay, same like DDBJ. It is a bioinformatics, and DDBJ Center provides sharing and analysis service for data from life science, research and advanced sciences. So you can get the detailed information, whatever you want to learn about it, you can get it over here. Okay. So this is, once I'll finish all that, I'll tell you what are the things you have to remember and collect from the data. If you want the handwritten notes, you can send a message in the comment section. I'll share the PDF for these notes, which will be useful for the writing part of purpose. Okay. So the first one is a nucleotide database. Second one is a protein sequence database. So there is a one database that is PIR, UniProt, and TREMBL. TREMBL is nothing new. It is just the sequences which are present in EMBL. It is the translated one. So translated means from DNA to RNA and protein. So all those DNA sequences which are present in MBL, the translated form of those sequences are present in TRMBL. Okay. UniProt is a universal protein sequence database, and it is UniProt KB. KB means knowledge base. Okay, the complete name of database. and it is the highly accessible database. Okay, nowadays PIR and all is it is up to dated, but all the information you can get it from the Uniprot in a easy manner. I'll show you just on this data. You can see here integrated protein information resource for genomic, proteomic, and systematic biology research. So if you want about the protein ontology, you can get it from here. See here, the universal protein resource provides a scientific community with a single centralized authoritative resource for protein sequence and functional information. So whatever the information which are present here, you can go get it from the Uniprot. So that's what I am saying. Like same from here, EMBL and DDBJ is little bit not public user handy. Like you cannot get the very easy manner. But in GenBank, you can get the data in a very just a single click. You can get it that information which is of your use. So that's why for nucleotide you can go to GenBank. Same in the case of protein, you can go to the Uniprot. So Uniprot contains the very much useful information. Every information, whatever present in PIR or TREMBL or SwissProt, there is a various protein database, but you can get it from the Uniprot very easily. Okay, so this is about the. You can just see on the Uniprot also like how Uniprot looks like. In this PIR, you can also find the integrated protein PTM resource. Literature, information, and knowledge. Other resources is all peptide search. If you want to use peptide, is a small segment of a protein. You can find it out. You can find out the text search. So that is about the pro class. So those information you can get it from here. It is maintained by the University of Delaware. Okay, that is in US. So when you will go to the Uniprot, Uniprot looks like this. So this is the fourth version released 2023 4. So it is very updated database. If you want about the proteins, you just have a one look. See, it is reviewed and unreviewed. It is mentioned here. So reviewed means there is an entry is 5,70,157 reviewed studies are present in this database, and 25 crores 16 lakhs 768 unreviewed entries also present. That's why it is name is a knowledge base. Means if something which is not tested in the lab and you have the sequence, you can submit in here, but you will not get the yellow star. It will be just in a white color. I'll show you just now. You can species, you can find the proteome of any species, protein clusters. You can find sequence archive is a unipath. You can find it out over here. Okay, there is a something in good time what is the latest news, you can find it out. Some tools like blast align search with list map IDs search peptides. This is online training resources is also available. You can take some training programs over here. Okay, and the downloaded version, and these things is available in the answer. Just have a look once and keep remember when I'll ask one thing in, you can answer in the comment section. So just I have typed here human, and you can see how many proteins are available. 66 lakhs 18,173. Okay, and how many are reviewed? 51,790. Just remember this number. Okay, 51,790. And here there is a one database. Just I'm opening it. I discuss about it. See here how many structures are available. 2,10,180. And just type human over here as I am searching just human data, and just click search. See the number, it will come. Just loading the data. See here 3,521, and it is something like related with human, or some it's not directly related with human, but let it keep it in mind. It is like all is related to human. 3,521. We have reviewed sequences 51,790, and we have sequence only 3,521. So simply meaning is, you just tell me in the comment section what is the reason for that, what why we have very less few structures. Okay, and I will post it in the comment section. Okay, it is just a homework task for you. Just go through it. Only just remember this number 51,790. Okay, or if you'll compare from this number 6,000 66 lakhs 18,173, then there is a very very less number 3,521. Okay, just post in the comment section what is the reason for that. Okay. So this is about the primary database, and what are the things you have to remember for this database section. I'll tell you just finishing after finishing these things. Okay. Second type of database is a secondary database. Secondary means there is a updation from primary to secondary. Means that data are some feature they have in that. So that data is a PDB. PDB stand for protein data bank, and it is maintained by the RSCB. Sorry, it is not RSCB, it is RCSB. Is the full form is Research Collaboratory Research of Structural Biology. Structural Biology. So, biology. Okay. So what is it? It contains the protein structure. Name itself, it's a protein data bank, but you can find some nucleotide data is also, some DNA structure is also available in PDB, but very few in comparative to the protein. Okay. You can find it out all the protein structure which are identified using the X-ray or NMR method. Mostly structures are identified from the X-ray crystallography method, like X-ray crystallography. I'll just show you how does it looks like. It is open here. Just when you will go to here, you will find a page like this. Okay, so it is a protein data bank RCSB. Total number of structure in PDB is 2,10,180. Computed structure models is 1,68,577 CSM. Okay, here it is a CSM is, it is not a crystallography identifier. It's a computer generated structure is available. So if you want to include CSM, you can add it here. If you don't want to include, you just click here. 3D structure, what you want, 3D structure or documentation. You can find some document also which is available in PDB database, but mostly we use it for the 3D structure. Just type your keyword, what you want to search. As I have searched here is a human. You can see here, this is the detail. Okay. Uh, it is mentioned some full text is a human structure attributes. You can make some using and condition, like human and this and this and this. So you can, if you have some very specific query, you can mention in this chemical similarity, structure, motif, anything, everything you can send it over here and you can identify. So you will get the data like this. Among the various identified structures, you can choose your mm, data which is of your interest. Okay, and you can very much understandable, like all the IDs are, it's a four letter. So PDB ID is just a four letter, and which is alphanumeric. It start from a number as well as from the some alphabet, but it is a four letter only. Okay, just look, when I'll talk about the PDB file format, I'll talk about the PDB file format, but here I'm just talking about the PDB. So once you click over here, it is mentioned structure of human serum albumin. It is a date of identification. It is mentioned here. Okay. Classification, it's a metal binding protein. Organism Homo sapiens. Expression system Coma Getalia pasteuris. Any mutation is found? No, there is a no mutation. Indicate sequence mutations if any available here. Deposited 9 March 2022. Released 10 March 2021. Okay. Depositions authors Xiang W. SUJ. Suje. Okay. Experimental data, as I told you, it's a X-ray. Mostly it is a X-ray crystallography. So it's a X-ray diffraction method. Resolution is a 2.4 angstrom. Resolution is a very important feature for this identified structure. If resolution is very low, means structure is not of good quality. If it is very much high, then also it is not for very good quality. So it must lie in the range of 2 to 5 angstrom for the better, for the best structures. R value free, R value work, R value observe. So this is just a resolution value in a different places. Here it's a WWPDB validation, like what is R free, clash score. So something that is about the, when you will perform the X-ray crystallography, you will get a graph. These colors are showing the energies. Okay. Here it is a 3D view, 1D, 3D view, electron density validation report. You can find it over here. If you want to read about the literature, it is not yet published. So once it will publish, it will available here. Here it is available some macromolecule. Protein is a macromolecule. You can find it out the macromolecular IDs. Okay, what is its serum albumin? And there are two chains. You can see here clearly it is two chains. So there is A and B. Sequence length is 585. Organism is a Homo sapiens. And there is a one thing I have missed. It, it is saying there is a no mutation, but there is a some sequential mutation is available. Okay, GIG2 0, GIG42. So there is a zero mutation, but the number of mutation in the polymer sequence that are engineered from the reference sequence. Okay, so there is some engineered sequence mutations are available. If you want to see, you can just click over here and you can read about it. This is the structure image. Come down, it is talk about the sequence clusters. Here it is the reference sequence. This is the reference sequence. When we modeled something, I'll come back here. I'll just little bit explain here about the what is reference. Just a second. What actually happen when we find out the structure? Okay, when suppose from the X-ray we find the structure, how you will find, you will get the best structure. So we have to some align, we have to perform some alignment, because in X-ray crystallography you are not just directly going to the shape of a structure. You will get a pattern, and from this pattern you have to identify the structure. So that's why it is alignment thing, it is we are discussing about. Okay, and then you will get the experimental data validation. There is a various length and angles are available here. Software, it's a Phoenix for refinement, and PDB extract for the data extraction. Okay, entry history, it's a release date, it's a, as I mentioned in the top, that is only. And there is one more very interesting thing, it's a version 1.0. So this is some, in some structures you will find various entry. Okay, version defines as 1.0 means it is the initial release. Once these authors, those who submitted the data, they will find there is a different update in this sequence, they will again update to the database. So there is a not a new entry for this particular protein structure. So there is an updation version 1.1, version 1.2, and there will be a date, and then there will be a, see here there is a no click because it is an initial release. Once there is a version, you can click here and you can differentiate what is the difference in the databases. Okay, oh sorry, not sorry, difference in the data. Okay, so that you can identify from the PDB. So this is the detail is about the PDB. And there is another database that is a PFAM. PFAM is a protein family database. The name of PFAM is changed to the interpro, okay, interrelation of the proteins. There is a different third protein database is a blocks, which is talk about the particular block. I think block is not available now. Okay, I couldn't find the link for the blocks. That's why I have given just a paper about it. That is in 2002. Okay, if you want to read about the how the block just works, you can read that paper. You can just click over here. I'll share this PPT if you want. You just ask in the comment section. I'll share this PPT also with you guys. Okay, so you can read about what is the database description, what is the recent development. I couldn't find there is any difference development after 2001. I couldn't find anything. It works on the llama algorithm. Okay, so suppose if you are going for some competitive exams, then you can uh, expect a question on such kind of things, like on what algorithm it is worked. So whatever things you can discuss, you can see in this page. Okay, so this is about the secondary database. You might be thinking like how to write the answers for these database. So I'll tell you, just I'll finish this, I'll tell you what are the things you have to remember for this writing part. Third one is a composite database. You also can say specialized database. So what is the different here? They have some feature. Feature means like they have a structure and we have some specific features in that particular structure. But in composite database, these databases are specifically meant for one particular purpose. Just for example, scope. Scope is a structural classification of protein. Suppose we have a protein family and that is lipase. Lipase is an enzyme. They have some structure. So what is the structural suppose one lipase and lipase mostly in this way. So there must be some slight differences. We can find it out. Okay. See, these three one have the same. Fourth one is approximately giving the same thing, but it is not same. So we can find out such particular differences. Why it is looking like lipase structure, but it is not a lipase. So that things you can find it out in a scope. Structural classification of protein. Structural classification of protein. Cath is a class, architecture, topology, and hierarchy. So suppose just click on the scope. How does it look like? It says scope 2, it's updated. Okay. Scope 1.75 and scope 72 prototype. The legacy scope website can be accessed at this. So means scope is get updated to this database. Nearly all proteins have structural similarities with other proteins and in some of these cases share a common evolutionary relationship. The scope database created by manual inspections and abated by a better battery of automated methods aims to provide a detailed and comprehensive description of the structural and evolutionary relationships between all proteins whose structure is known. Okay. As such, it provides a broad survey of all known protein folds, detailed information about the close relatives of any particular protein, and a framework for future research and classification. It is lastly updated on 29th of June 2022. Includes 7254 non-redundant domains. Non-redundant means which is not repetitive, representing 8,61,631 protein structures, folds, superfamilies, and families. Statistics here. You can just click here, you can get the statistics. You can go for alpha proteins, beta proteins, alpha and beta proteins, small proteins. See, there is a difference. Alpha and beta, A slash B, A plus B. So means added and along with. Okay. Globular protein, everything you can get it here. It's a wonderful database. You can search, just click anything and you can search over here. Second one is a cath. Just click here. You will get about the cath. 151 million protein domains classified into 5,841 superfamilies. Okay. Core classification file for the latest version of Cath Plus. It's now available to download. You can download. Protein evolution, you can find it out. Protein function, you can find it out. Conserve site, that is very much important. Okay, and 3D structure. Find out the 3D structure your protein adopts. Means we have one structure, and what is the possibility your structure could have the what kind of folds it can have, so that even you can find it out from the cath. What is cath gene 3D? There is a different thing. There is a release of cath plus 4.3.0, that's updated version. Okay, some tweets are there for cath, citing the resources funding what from the NIH, Wellcome Trust, ERC, MRC, Medical Research Council, BBSRC, Bioscience for the Future. So these are the funding resources. You can get it from here. Is a part of the Elixir infrastructure. So there is a something new they have launched. You can check it over here by clicking search cath by text or text ID. You can find it over here. It's a wonderful database. These two, like a scope and cath. If you are working and really looking for some project and all something in the field of protein, okay, you must have the knowledge about these two particular databases. Okay. Now third one is a CAG. That is also a very important and wonderful database. CAG stand for the it's a Kyoto Encyclopedia for gene and genome. It is meant Kyoto defined as a Japanese database. Okay, so it is like a uh, made by the maintained in the Kyoto University. That's why it is a Kyoto Encyclopedia. Encyclopedia means which contains the detailed information. Like it contains a map. If you'll go to the encyclopedia, you open it, you will find some maps. So by understanding the function of gene and a genome by the map, I'll show you just how it is looks like. Okay, it's a pathway database. See, Kyoto Encyclopedia of Gene and Genomes. CAG is a database resource for understanding high-level function and utilities of the biological system, such as the cell, organism, and ecosystem, from molecular level information, especially large-scale molecular data sets generated by genome sequencing and other high-throughput experimental technologies. Okay, it is updated on August 1, 2023 is a highly updated database for new and updated resource. So what actually happened? Uh, just check here. I'll just show you. No entry form. Why? Why it is not showing any entry? Uh, See, just have given glycolysis. So everyone have gone through the glycolysis pathway. How does it looks like? See, this is the complete pathway. What we studied in our classroom, just in a 4, 7, or 8, 10 steps. It's not in the 10 steps. It's such a healthy process. You can see here. So this number it is known as a EC number, enzyme commission number. Each enzyme has a particular number and you can see how typical the pathway is, how does it looks like. It passes through how many steps. Okay. So it is a glycolysis and gluconeogenesis. Both pathways are here you can see. So CAG is a wonderful database when you are going to it was showing some typo error or something. That's why it is what I was looking before. It is not showing, but you can find it out about any pathway which is running inside uh, our body. Okay, so most of the pathway you can find it out in the CAG. It is available in the this particular database, and it is very interesting database to understand the biological function. How does it behave, and what is the possibility? Or if you have some disease, and that disease pathway is also available here, you can find it out, and you can understand how to control the disease. At what particular gene we should work to control this particular disease. Okay, so that's why it is a very important and very good database. Third one, next one is a Brenda is also an enzyme pathway database. Okay, it is also a good database. So this is Brenda. We have migrated some of our central management database to a new services. So database migration has occurred in September 27th. See, it's just a 4 days before release is there. So it is also highly updated. It is released 1st February 2023. So you can go for the structure based query, you can go for predictions, you can go for visualization. So this is information, enzymes and ligand, organisms, activating compound, amino acid sequence. So such information you can find it out from Brenda. Also you can find out some enzyme history and all, which is included in the Brenda database. Next one is a tiger. Don't go come confused with the tiger which is in zoo. So it is a small t. This ti stand for tissue. Okay, so it's a tissue specific gene expression and regulation. Tiger is a database developed by the Bioinformatics lab at Wilmer Eye Institute of Johns Hopkins University. The database contains tissue specific gene expression profile or expressed sequence tag EST data, cis regulatory module CRM, and combinatorial gene regulatory data about the database. So you can read about lots of things over here, and it is tissue specific. We have lots of tissues, and every tissue have some particular genes and how they get expressed and how they get regulated. So like suppose there is a one lac operon, you have must have read about it. So one particular gene can maintain the complete function, and there is a combination of genes they may also maintain a function. So like that only in a tissue, Like suppose if a tissue is functioning X function, so that X function is working by self or it is involved, there is a variation of genes. You can find it out from this particular database. Okay, so that is tissue specific gene expression and regulation. Then there is a panther. Again, this is not the same panther, but the logo is same. Just you will see here. Uh, See, this is the same Panther, but it is not that Panther. So the mission of Panther Knowledge Base is to support biomedical and other research by providing comprehensive information about the evolution of protein coding gene family. Protein coding gene family, as I mentioned here, just have a look. As I was saying that from DNA sequence, if suppose it is a human, it is a 3 into 10 to power 9 base pair, and from here we have only 3%, 2 to 3% of the protein coding genes. So, how the evolution has occurred and how a particular gene is functioning and how it is making a protein, that information you can get it from the Panther database. You can find you just enter the IDs here or you just certain information you can gather here. You can get about the information. So it is also a good database. Okay, you can read about it. Gene ontology, Geo, GEO. Okay, so it is a gene expression ontology, gene expression omnibus. Okay, it is a part of NCBI. This database is a public functional genomic data repository supporting Miami compliant data submission, array and the sequence based data are accepted. Tools are provided to help users query and download experiments and curated gene expression profile. So if you want to read about the profile of a particular gene, you can use this particular tool from the NCBI. You can identify. Then there is a OMIM. It is also a very good database. OMIM stands for Online Mendelian Inheritance in Man. There is a OMIA, that is Online Mendelian Inheritance Animals. Okay, and here is a man. OMIM is supported by a grant from NHGRI, license fees and generous contribution from people like you. Okay, so they are asking for some donation. Yes. So if you want to search something, okay, so just for example, I'll show you how this looks like. So it's a basic search and there is certain operators. There is a phrase, wildcard, field search, Boolean operator. So these things you can search over here and any clinical features, suppose Down syndrome, just for example, Down syndrome. And you will click here, see everything comes here and now you can see here it's a text description, clinical features, diagnosis, cytogenetics. So any genetical disease you can read here, and there is a one gene map you will also find in some places like here. See, just a bit it will open. I'll show you. See, this is the complete map of the gene. How does it looks like? Okay, and this is the complete chromosome. We all know Down syndrome is on the chromosome 21. And what actually happen in chromosome? It's a trisomy. Okay, so the position, actual position, if we want to know, it's a chromosome 21, Q 22.2. So what is this Q is? You just understand like this. Each chromosome have suppose there is a one chromosome. This is a chromosome. So one is a small arm and one is a large arm. So chromosome have a small arm is defined as P and large arm is defined as Q. So अगर आपको ये याद रखने में प्रॉब्लम तो ऐसे रखिए कि Q को बनाने में ज़्यादा पेन चलाना पड़ता है. So you require to move larger walk long to make a Q. So larger Q. So 21 Q 22.2. So it means chromosome 21, larger arm, position 22.2. So इसी तरीके से अगर आप किसी भी जेनेटिक डिसऑर्डर के बारे में जानना चाहते हैं, so you can find it out about from the YIM database. So this is about the detailed description about the biological databases. You can find it out about, all links are available. Just click me, just send me a message in uh, comment section. I'll share this with you, your details. I will send it. So as I told you before, what you have to write, you cannot learn the database. It is updating daily. So whatever the things you have to remember, first one is the introduction. You have to mention about it. And introduction includes uh, description of the database, description of database. Okay. It also includes uh, the funding agencies for who is funding for this, what the database is doing, okay, function of database. So this part is an introduction, okay, and then you will come to the uh, classifications, like what actually it is, a nucleotide, it is a protein, okay, what types of data is present in this database, what type of database, okay, and how the data comes from, like how data receive data entry, how to enter the data, how to retrieve the data, and the flat file. So these basic things you have to write about the particular data. Flat files I will cover when I'll discuss about the flat files in just the next lecture. I'll talk about this. So these things you have to mention it. And better thing, if you want to learn in detail about the database, just go to the nucleic acid research, nucleic acid research, and here you will, sorry, and here you will find about the archive. When you will go here, you will find two database. There is a January and there is a July. There is a two database. They are specific, one for tool and one for databases. So you select the database one, go there, type the database name whichever you want to study. Okay, you will get the updated paper. Okay, nucleic acid research is a journal. In that you will find the updated updates about that particular database. If you want to just the basic, read the old versions, like 5 years back or 6 years back. So you will get the general information. Okay, from there you can prepare your notes. And if you do not want to prepare your notes, just send me your details in the comment section. Okay, I'll share the notes with you. You can study from here. So just to conclude, in this lecture, what we have covered about sorry, sorry, we have covered about the basic about the bioinformatics, what is bioinformatics is, how does it looks like, what is databases, what is biological database, what is the importance, and we have discussed about the various types of databases, that is primary database, secondary database, and composite database. Any doubt, any discussion, if you want, you just make a comment in the comment section. Okay, I'll revert back to you. If you want to get notes, just mention your in the comment section about your mail ID, and also I can share the notes with you. Okay, or any kind of comments, any information, any discussion you want to make, you can make comments in the comment section. You can read it, I can read it and revert to you. Okay, like the channel, subscribe to my channel, share with your classmates. Okay, and surely you will enjoy while learning when we'll move apart through the syllabus. Okay, thank you very much.